Okay, so now that we have our mRNA transcript, and now that it's floating outside in the cell's cytoplasm, and let's just make a little picture right here. Here's a cell, here's its nucleus, and what's happening is we've got a little mRNA kind of floating outside in the cytoplasm, and the mRNA is looking for a ribosome, because a ribosome is where translation happens. Let's draw our mRNA transcript here, and note that I'm going to have our mRNA transcript go from the 3' end on the left to the 5' end on the right. This was not the way that I had it on the transcription slide. If you go back to the transcription slide that goes along with this, you'll see that on the transcription slide, it was left to right, 5 to 3. Now, you can always tell where the start of transcription is because the first sequence of 3 nucleotides on an mRNA is always AUG. In fact, a sequence of nucleotides, 3 nucleotides, on mRNA is called a codon. So that means this is a codon. It's the second one. This is a codon. It's the third one. And so on and so on. And filling these in from our mRNA transcript that we made, we can see that we have 5 codons here. So in order for translation to start, a small ribosomal subunit needs to dock. Oh, let me draw that better. A small ribosomal subunit needs to dock on the mRNA. In fact, ribosomes don't exist as a complete ribosomes. They actually float around as the small and the large subunit. They only come together when it's time to do translation. The other thing that has to happen is not just the small ribosome subunit has to dock. We also have to dock the first tRNA that is also carrying the first amino acid. And because AUG is always the start codon to mRNA, that also means that this amino acid called methionine is always the first amino acid. Now, not every protein retains methionine. Some of them will cleave it off before they get folded. This tRNA, it's got an amino acid attached to that side of it, but it also has a sequence of 3 nucleotides, and that sequence of 3 nucleotides is called the anti-codon. And it uses the base pairing rules to see what 3 nucleotides are actually on it. And as you can see, there are other tRNAs that are floating out around that I'm going to use in the making of this specific protein. And let's just look at these tRNAs for a minute. One of them is going to be a tRNA with the anticodon CGG, which seems to match one of our codons. We have another one that's going to have an anticodon of CCA, which also seems to match one of our codons. And I have one here, AUA, which also matches. I'm not going to draw one for this very last codon because this last codon here is what is called a stop codon. When the ribosome reaches this codon, it'll finish translation. It's just like, I'm out. I'm done. I'm out. Now, each one of these tRNAs does have its own amino acid attached to it. And you might say, well, which amino acid? That is set in stone. There's actually something that's called the genetic code, and what it is a decoder ring. It matches an amino acid with its codon, and this is what the genetic code helps us figure out. We can use the genetic code And we can find these codons on the genetic code, and we can also find what amino acid goes with them. So the genetic code is a very powerful decoder ring, basically. Before we even get started using the genetic code or anything like that, now that we have the small ribosomal subunit docked on the mRNA, and now that we've docked the first tRNA and the first amino acid, the large ribosomal subunit then docks itself on this whole machinery here that we have. And let's label this. This is the large ribosomal subunit. The large ribosomal subunit actually has docking stations on it. And these docking stations are very much like the garages at a gas station. Like there's one docking station here, there's one docking station here, and there's one docking station here. And as you note, I put methionine in the middle docking station. The docking stations have letters that go along with them. This one here is called E. The E is for exit. Any tRNA in the E site will actually be exiting without its amino acid. This is called the P site, and this one here is called the A site. Those two names are named for very complex chemical reactions. Basically, what you should know is that if there are two tRNAs in the P and the A site, their amino acids are going to get fused together with a peptide covalent bond. I am going to stop this video here, and then I'm going to start another video that shows us going through the steps of translation.